Welcome to the latest Novogene webinar, Introduction to Novogene's Clinical Sequencing Services. Today's webinar is different to our usual beginner's guide. We will focus on Novogene's clinical sequencing services, discuss the clinical pipeline flow, clinical WES and WGS services, clinical panels and sample requirements. You will find out more the details during the webinar. A bit of housekeeping before we start. The presentation will run for around 40 minutes with 15 minutes for question and answers at the end. Please submit your questions via the Q&A button at the bottom of your screen. You can upvote a question by clicking the thumbs up symbol next to it. We will try and prioritize the most popular questions. If we don't have time to answer your questions, we will come back to you via email after the webinar. Please don't use the raise hand functionality. Just send any questions via the Q&A or message us directly if you have a technical issue. We will share the slide deck and a recording of the webinar via email. You can expect this on Friday. If you are studying and need a certificate of attendance, there will be a link in the follow-up email that will display the certificate you can print from your browser. Finally, there will be a short survey that pops up when the webinar finishes. Please take a couple of minutes to complete this, as it helps us to plan future webinar topics. I would now like to introduce your host for the webinar, our scientific content specialist, Eva. Eva, over to you.
Thanks, Miranda. Hello, everybody, and thank you for joining our webinar today, introducing Novogene's clinical services. My name is Aoife, and I am Novogene's Scientific Content Specialist and your host for today's webinar. Shortly, I'll pass you over to Ina, who is our Clinical Technical Support Manager, and there is no better person to take you through our range of clinical sequencing services. Before I do that, I would like to speak a little bit about Novogene, just for anybody who's joining us here today for the first time. So Novogene was founded in 2011 and has been growing rapidly ever since then. Our company mission is to advance genomics and improve life. And as you can see here, our services range from complex disease research and oncology to food and water safety, and cover a broad range of real life applications. Our European Sequencing Centre is based in the Cambridge Science Park in the UK. Here we have our ISO 17025 certified state-of-the-art lab equipped with the latest Illumina NovaSeq 6000 sequencing machines. We have always provided fast turnaround times and high quality data, but that has improved even further with the recent installation of Falcon 2, which is our new intelligent sequencing platform and has increased our monthly capacity by about 200% compared to manual steps. We also have our supercomputing clusters, so we can offer bioinformatics services and store your data in the UK. However, at our heart, we are a global company and we can offer services across Asia and the US and, of course, Europe. Falcon 2 has been deployed across a number of our global labs, which has standardized our services. We've got almost 2,000 employees and we serve over 5,600 customers in world-class universities, biotech companies and hospitals. And we can provide sequencing for the smallest to the largest of projects. We provide a full range of next-generation sequencing services, such as whole genome sequencing, whole exome sequencing, various types of RNA sequencing, metagenomic services, and epigenomic services. These can be applied to a wide range of research topics, from cancer and rare disease, microbial cell factories, and gut health, to agriculture and animal health. The focus of today's webinar, of course, is on our clinical services. So Ina will go into much more detail on these shortly, but briefly, we have our NovaPM 2.0, which analyzes 484 cancer-related genes for alterations at the DNA level. Our NovaFocus, which analyzes hereditary cancer risk. We also offer targeted sequencing panels based on the disease of interest, and these are customizable. We provide clinical grade whole genome sequencing and whole exome sequencing with our Agilent clinical whole exome service accredited by ISO 17025. Our services can be applied across a wide range of health-related areas, such as carrier testing, cancer diagnostics and monitoring, and pharmacogenetics. Many other companies offer these services too. The NGS market is quite crowded, so why should you choose Novogene? I think there are plenty of reasons why you should go with Novogene for your sequencing project. Our Cambridge Sequencing Centre is ISO certified, guaranteeing you a clinical grade standard of service and data quality and some of the fastest turnaround times in the industry. As I mentioned, we've had a really exciting development with the installation of Novogene Falcon 2, our NGS, our intelligent NGS delivery platform. This has been deployed across our global lab network and has standardized our services. This technology was developed in-house by Novogene and really highlights the innovation that we strive for at Novogene to keep pushing the bounds of the genomics industry. We also have one of the highest sequencing capacities in the world, and that is set to improve quite soon with the upcoming installation of the new Illumina NovaSeq X Plus at our Cambridge Sequencing Centre. The PacBio Revio will also be introduced to our Tianjin lab in China and will greatly increase our capacity for our long-read sequencing services. So keep an eye out on our social media over the coming months for further developments on this. So the benefits of our in-house innovation are clear in our record in the scientific community, and we're quite proud of that. We've been cited in over 790 research papers with a total impact factor of over 6,120. We're also driving the NGS industry forward with 37 NGS related patents. Our technology is helping to make the NGS process more efficient and effective, all the while improving the quality of the data we deliver. The Novogene community keeps on growing. Our experts sales team speaks over 10 languages and is located across Europe, ready to assist you with your next project. If you're not ready to send us samples just yet, you can still benefit from our educational outreach to learn how NGS can work for your project. We've recently kicked off our 2023 Roadshow seminar series, starting in the Netherlands, Brussels, and Italy. If you would like us to organise a seminar near you, please do not hesitate to get in touch. You can also keep an eye out on our social media for the announcement of dates already organised in your city or your institute. I would like to quickly introduce you to our European sales team. They are, as I mentioned, based across the continent and ready and waiting to help you with your next sequencing project. If you would like to speak to one of them, please feel free to email us at info@novogene-europe, and we will put you in touch with your area representative. As today is focusing on our clinical services, I would also like to introduce our fantastic clinical technical support team. So we have our clinical TS manager, Ina, who you'll meet shortly, and her team, Sonia, Paula, Robin, and Sergio. So with that, I would like to thank you for your attention, and I will hand over to you, Ina.
Thank you, Aoife, for this nice introduction. I'll proceed to, in a second, share my screen. I hope everyone can see it now. So, well, welcome everyone, and thank you again, Aoife, for this nice introduction. So this is Ina, Clinical Technical Support Manager at Novogene, and today I'm going to introduce briefly to our clinical services that we can offer. So basically, here as an overview, I'll give you a brief introduction, followed by explaining a bit our clinical pipeline, the clinical services, including whole genome and whole exome sequencing, our panels, transcriptomic solutions, and also briefly in the end, some solutions such as pre-made library and long reads. So basically, to give you like a little bit of context at the beginning, so I would like to highlight that rare diseases quite affects like our population, being, for instance, in the European EU population, from 6 to 8% of them. So this also implies like a important burden in the health system. And however, like 80% of the rare diseases, it's known to be having like a genetic origin. And also to adding to that, we know that more than 200 million people like worldwide are living with an unresolved genetic disease. And this could be even higher due to the fact that like some of them are not really recognized yet. So therefore, like this is why like NGS has a really important role into here, because it has had like a great like development in the recent years and transforming genetic disease diagnostics in like a huge way, giving high throughput and a cost-effective solution for these diagnostics, preventing unnecessary invasive procedures, also allowing like an enhanced surveillance and a better diagnose for like the diseases, like the disease management, prognosis expectations, and also adjust and change any medication or dietary like requirements that the patients might need, so that like we can have like improvement of the treatment of care. Also, not only in rare diseases, but also NGS has been paramount into cancer, providing a more sensitive, accurate, and fast turnaround time for samples that are like really precious and sometimes cannot be reproduced again. Therefore, NGS means like a way forward to personalized medicine with like the ability to be detecting like driver mutations, resistance mechanisms, germline mutations, and quantifications of biomarkers such as tumor mutational burden or MSI, and therefore improve the patient care as an outcome. Finally, I just want to briefly highlight that up until now, for the clinical diagnostics, Sanger sequencing has been like the gold standard. However, with like all the developments for NGS, we can really say that NGS has many advantages, including comprehensive genomic coverage, higher capacity with sample multiplexing, lower the limit of detection, faster turnaround time for like a huge volume of samples, and also just like for conclusion, I just want to say that therefore we can conclude that it's like a very time and cost-effective solution. So at Novogene, we want to be like your partners into that, and therefore take your sequencing projects with us to be able to support you in all the fields that might be included in diagnostics and care. This can be from predictive and pre-symptomatic medicine applications on pharmacogenetics, all the way around pregnancy, from carrier testing to pre-implantational testing, or prenatal and newborn screening, and of course for the genetic disease diagnostics, gene expression profiling, and all aspects into oncology and cancer, such as risk assessment, diagnostics and monitoring, and finally also in aspects like consumer health, that it's also a part that is growing as population have higher interest into their well-being and fitness in general. So to do that, at Novogene, we have different accredited labs. So we have medical testing laboratories that are accredited with the CLIA laboratory improvement amendments, and also the College of American Pathologists, which both of them focusing, providing high quality data and ensuring that the results we provide and the data we provide to you are high to the field standards. In Europe, as Aoife was mentioning before, we have an accredited lab in Cambridge, UK, and it has the ISO 17025 that again, it assures the high quality of the data and a quality management system being implemented, and therefore all the steps that follow at the lab ensure that you would get a high quality data, and therefore fit to use for your further analysis and diagnostics. We also have GCLP, which stands for clinical laboratory implementation, and it's especially relevant for clinical trials and clinical research that also needs to prove that it's really good, well performed. So this clinical laboratory practice is really relevant certification for this kind of applications. And finally, you have the HTA license, which it's a requirement for using and storing tissues from human origin. And having this license implies that we are processing the tissues in an ethical and way, and illegally having all the consent from the patients that provide the samples to us. So moving now to the next step, which is the clinical pipeline. So you can see here briefly aligned how the pipeline looks from the extraction, in case that you would be sending us tissues to us, until the end, to the analysis that we would provide to you in case that your project requires that. But mainly what I want to highlight from the clinical pipeline, it's certain points that are paramount for providing high quality data and reliable results, which is that all the experiments in our wet lab are done by licensed personnel in certified laboratories, and every step is double checked, providing even further reassurance. Also, when we provide the report to our customers, it's always reviewed by our team of experts as technical support, and also signed by our bioinformatics engineer. In addition to that, we always have an internal quality control for the clinical samples before releasing, checking against sex identification, comparing with the initial information provided by our customers in the sample information form. And anytime there is any detection of a mismatch into that, an immediate troubleshooting process will be started, and an investigational report will be generated troubleshooting this any issues on that. And finally, want to emphasize that there's a regular vertical check done by the lab manager, which also adds another assurance into that the processes are performed rightly. So now moving specifically step by step. So in our laboratories, we accept both already extracted DNA or RNA, but also tissues, and we can do the extraction for you. So the main tissues we accept are like blood, buccal swabs, saliva, or FFPE. These are the main tissues we accept, but it's like our range doesn't are not limited to that. So we can also accept any other tissues that you would like to send to us, but we would go first through an evaluation and then recommend you the best way to send it to us in terms of the collection and transportation recommendations, as you can see here, for instance, for some of the tissues I was mentioning. After that, we would perform the extraction and do the sample quality control. So this quality control includes both qualitative and quantitative checks. That includes the volume measurement, concentration by qubit, and electrophoresis gel to assess the degradation or the integrity of the samples. Once this is done, we would generate a report that will be shared with our customers and with informed information for situation, so that the customers can take a decision on how to move forward with the samples. And once the customers confirm to us how to proceed, we would then prepare the library for each of the sample, considering a specific protocol for each of our services, so that we can provide the best outcome to that. And after that, we would perform the library quality control. This is an internal check that we would be also again checking on concentration and performing qPCR and profiling of the library to ensure that the library is fit for sequencing. Once this is done, this internal control would be proceeding to the sequencing with our NovoSeq 6000 machines. And after that, a third quality control will be performed onto the data. In this case, what we would be doing is remove any low quality reads or reads containing uncertain reads into that and in certain bases. And finally, also any adapter contamination, so that after that, if any further analysis is required, we would only use the clean data to do the rest of the analysis. So at this point, we could either deliver you the raw data or proceed with any further analysis you might require. And related to analysis, I will be talking further later on my talk. Now I will move specifically to the clinical services we offer. As I mentioned before, we have a wide range. So I will start for the first one, which is the whole genome whole exome sequencing. And let me first share with you this slide that compares different strategies. So first of all, you can see the whole genome sequencing, in which when performing this service, we are interrogating against the whole human genome, and they're based on the reference genome, and therefore all the kind of variants can be detected, including more complex and structural variants, and also identification for the non-coding variants, non-coding RNAs or mRNA splicing. And this strategy also is quite useful in terms of being able to be rechecking and reanalyzing across multiple variants, increasing the diagnostic efficiency. However, for a most cost-effective method to check a genetic disease and be able to provide the diagnostics, there's whole exome sequencing, which by targeting only approximately the 2% of the genome, which is the exome that encodes for most disease known variants, it's a very widespread used method. So why this is used? Because more than 85% of the disease-related mutations are found in this area, and therefore, as I was saying before, this is a very popular method as a cost-effective one. Finally, we have the targeted region sequencing. At this one, it would be focusing in a much reduced area of the genome, targeting only a handful of genes that our customers, the companies interested into this diagnostic is specifically targeting a set of genes that are already known to be relevant for that specific disease. This, as I was saying, gets a more reduced targeted area, but can give you higher depth, and then be also a very cost-effective solution for your projects. And so at Novogene, we can offer this TRS or also our oncology panels. Now giving you more specific details onto our whole genome sequencing. So we provide high flexibility in terms of like data output and coverage, going from low pass to the classical 30x, 50x, etc. And also very fast turnaround time of 13 days from sample receival to the release of the data to you. And aside from being able to provide just the raw data, we can also like add some analysis and provide some customized deliverables such as the mapping files, spam files, variant calling files, and annotation to you. And always, like for the delivery, we would be providing a safe and personalized data transfer, either using AWS, encrypted hard disk, server transfer, or any other options that you might require, like with a prior evaluation on our site. And finally, I want to highlight like that we have a very high quality data production, and with like when we sequence, for example, like over 90 gigabases, we get like a 30x depth with the coverage of 10x being over 98.5% and a Q30 value over 85%. As I was mentioning before, I want to just like give you like a slightly in-depth information on what is low pass. So I said before, like we can do like the classic 30x sequencing, but also sometimes low pass whole genome sequencing or shallow sequencing can be a very interesting alternative to genotyping, because so in this case we would be like sequencing like low data per sample, but this complemented to statistical imputation can be a very powerful solution, and it has many applications into included into pharmacogenomics, genome-wide association studies, or also to increase the representation of variants into like minorities populations. So now moving to the next product, like whole exome sequencing. Here we can also provide like flexible data output and coverage, and our turnaround time is 10 days from sample receival to data release. We can provide you like multiple enrichment possibilities, so including Agilent, IDT, and Twist, and I will talk about the specific kits we have in the next slides. Also, again, here for whole exome sequencing, we can provide customized deliverables and safe and personalized data transfer. So for Agilent, here you have some further statistics. So we use Agilent Short Select Human or exon B6, and you can see that by sequencing 12 gigabases, we can provide a mean sequencing depth of over 100x with Q30 value of over 85% in terms of quality, and that the repeatability and sensitivity and specificity for both SNBs and indels are 95, over 92, and 99% respectively. So this ensures like we are providing a very high quality data. And also if here, like if you require like analysis, I was saying we have like the options of providing standard analysis and some advanced analysis. The standard analysis for both whole genome and whole exome sequencing would follow this similar pipeline. That would go from raw data, and after doing the data QC, we would obtain the clean data, which will then align to our reference genome. And then like be able to do the variant calling for SNPs and indels for the germline. However, if you provide like tumor and like a normal sample paired, we will be able to be calling also for SNPs, indels, and CNVs. In the case of the whole genome sequencing, we could add here as well like the calling for the structural variants. And of course, finally, we provide like the annotation. For the case of like further analysis like needed in advanced analysis, we provide solutions for both like disease and cancer situations. So for instance, for disease, we could add like solutions such as like candidate variant filtration, linkage analysis, or region of homogeneity analysis. And for cancer, further analysis in the driver gene analysis, mutation spectrum and signature, or screening for predisposing genes, or TMB calculation and neoantigen identification, which are like quite relevant for us as markers in cancer. And also as a globally personalized analysis, we can also provide results into xenograft tumor analysis or HLA tapping, which has been like very relevant into like the field of organ transplantation, population evolution, gene therapy, or in immunological diseases and cancer. Now moving to the next solution for whole exome sequencing, we have IDT XGMB1, which is a panel which is like a smaller targeted region. And here also I want to highlight that by default, we also include the mitochondrial DNA spiking for all the services we provide with this strategy. So in this case, with like 10 gigabases of data, we obtain over 100x of mean sequencing depth and over like 5,000x for the mitochondrial DNA. Also, the data quality is over 85% of the metrics of like Q30, and the bases covered are over 95% at 20x, and 100% of 1000x for the mitochondrial DNA targeted region. Now moving to the latest addition to our solutions for whole exome sequencing, we have Twist Exome V2. This one is quite recent, and it's like 36.5 megabases of the total targets, and especially designed like against like the reference genome AG38, and like improved for like a compatibility with like Nova 6000. The content of this whole exome sequencing kit contains like the sequencing in all major like gene databases such as GeneCode, RefSeq, CDS, Ensemble, and also covers like fully like UCSC and RefGene, and some other like so the cosmic pathogenic variants, ACMG 70 genes at least, and also related to the non-coding and other content. They contain also like all pathogenic variants, unlikely pathogenic for ClinVar, and many other like databases. Also, like to highlight that it also contains like the SNPs with targets, including like the data sets of Pengeli and Urogentes that has been like of reference in the field. Now moving to the next part is like our TRS and panels services. So targeted sequencing, it includes for like the small number of genes, and as I was saying, would be suitable for when you are like interested in targeted region smaller than 500K, that by sequencing around 1 gigabase would give you like a depth over 200x. And we can provide Novogene like optional analysis into that, providing like SMP and an indel calling. Into like now moving to the oncology panels, we have a couple of solutions, which is NovoFocus CR and NovoPM 2.0. The first one, NovoFocus, it's focused on hereditary cancers, and it includes 106 genes that are associated with the main cancers such as prostate, breast cancer, ovary cancer, etc. And by sequencing 1 gigabase of data, you can already get like over 600x of depth. Also, in this, we can provide optional analysis, which can derive like with the reporting of a technical report and like interpretation report, depending on your preference. And finally, now moving to the NovoPM 2.0 panel, this one is like our most popular panel that gives like a very important, like a relevant solution into the field by analyzing 484 genes for clinical important alterations, including indels, SMPs, CMVs, and fusions and structural variants. And this is specially designed for solid tumors. And I want to highlight into this that we are very proud of this product because, as you can see in the image below, not only like can be calling and contains all the genes included in Foundation 1, CDX, that it's like highly recognized in the field, but also many more. And it has been recognized that like the genes contained into this panel are like of relevance for like diagnostic in cancer based on medical literature and also the NCCN. Finally, for here, I want to highlight that this is this panel is performed in our CAP-accredited lab, as well as like our NovoFocus one, and the data is analyzed by our in-house bioinformatics specialist. For NovoPM 2.0, the depth is really high with over 1,200x of the targeted region. So giving you a little bit more of insight on the NovoPM 2.0, as I was saying, it gives you like very high coverage onto the clinical relevant genes, this 484. The turnaround time for that, it's 15 days from sample receival to data release. And for the analysis, we can always like include the BAMP, PCF annotation, but also information on tumor mutational burden and microsatellite instability. And as usual, like the delivery of the data is safe and personalized according to your requirements, and we provide you like this high quality data that I will go into more details in the next slides. Again, like this NovoPM 2.0 panel is based on like solid tumors, and therefore like can give you like some answers and to different applications such as targeted therapies, immunotherapies, and assessing like the cancer risk and efficacy of chemotherapies. So related of the quality of the data and the validity of this panel, I just also want to highlight that very importantly we checked against like reference standards and real clinical samples. So with an average sequencing depth of over 1,400x, we could see that the accuracy of both SNVs and indels, the detection was 99.5 and 93.7% respectively, with variance of minor allele frequency over 1%. And also like while checking some biomarkers such as like the CMV for HER2, it was like 100% when the tumor cell content was over 20%, and for the fusion of ROS1 unknown, it was also getting like 100% and very highly reproducible when the tumor cell content is over 20%. And in relationship with like the real like the clinical samples, we analyzed 41 like FFPE samples with different like conditions, ranging from known prior mutations to hotspots in SMBs and indels, and also like hotspots in fusions. And after like sequencing and having an average sequencing depth over 1,400x, the result is that like for the detection of SMBs and fusion, it was 100% concordant with the NMPA approved CDX test. So I'm mentioning this one because this test had been like previously approved by the CFDA, and therefore like the concordance is highly relevant as like an important like panel for like for NovoPM. Yeah, now I want to highlight the importance of like the variability on like the different kind of samples we can accept. So we can accept from your side like either like tissue or the already extracted like genomic DNA, FFP derived DNA, which many of the clinical samples, like especially the ones in archives, are derived from, and also like circular DNA, so CT or CF DNA. Here you have like some recommendations and suggestions in terms of like amount and quality, and like the collection and transportation. Mainly like what it's quite key here is like to highlight like the importance of like the samples coming from FFP are not too degraded, and that like the CT cfDNA doesn't have like genomic pollution. Like after that, I want to highlight also for this kind of samples that we have some recommended sequencing, and for genomic and FFP DNA, our effective to get like an effective coverage of in between 1,200 to 1,400, we recommend 12 gigabases of data, while for like a CT or cfDNA, it's 16 gigabases that gives you in between 1,400 to 1,600. And then as a control reference, only by sequencing 1 gigabase, you would get like 100x. And this data is like the one it's used as reference, so that is the baseline to be able then to be calling for the somatic mutations. Here I'll go like now to the pipeline for the analysis of the NovoPM. So as previously mentioned, like was after we get like the raw data, we would perform the data quality control and get the clean data, which then we will be mapping against the reference genome. And once we get like normal samples only, we'll be able to be calling for SMB and indels. But when paired samples are sent, we call also for somatic as SMB indels, CMVs, and fusions. Also, two other like parameters that we can calculate is the MSI and TMB. So to go further into this, like you probably are like familiar with these concepts already, but just like to make sure that everyone is on the same page. So the tumor mutational burden is the total number of mutations per coding area, and in the case of like our panel, would be based on like a calculation on the coding DNA sequencing sequence that it's approximately 1.4 megabases. And like the importance of these values, because it's been like found like to be a relevant biomarker and correlating with like efficacy of like certain immunotherapies for like many tumor types. And then like same for like microsatellite instability. So this one is the predisposition of mutation, or what is called like the genetic hypermutability, and that results from this impaired DNA mismatch repair. And this microsatellite instability has been used as a marker for like Lynch syndrome diagnostics, and also to do some prognosis prediction into like the treatment of different like cancers, and again like as a like a correlating for like immunotherapy agents as I just go through the Anotivo. Finally, I just want to mention that like the software we use for like a tumor sample only for like microsatellite instability assessment is M6. So here, as I was saying, like our like the NovoPM panel, like the findings in terms of like the variant calling can be used many times as biomarkers associated with FDA-approved therapies. So I was just now mentioning about like the MSI and TMB, and here you can see in this table that both of them, when they are found to be affected, they're like can be linked into FDA-approved therapies such as Keytruda or Opdivo, but also like in many other cases, such as in ovarian cancer, when BRCA1 or 2 are affected, then we could be associating with like olaparib or rucaparib as like a treatment. Or like finally also one last example in terms of like non-small cell lung cancer, when ROS1 fusions are found, crizotinib would be like the recommendation in terms of like therapies and drugs to be used. So here, just to give you like even like more like clear view on like how this panel is like useful. So there's this case of like non-small cell lung cancer that after getting like through like this NovoPM panel sequencing, the diagnostic result was like a finding of a novel ROS1 fusion, and therefore it was associated, as I was mentioning before, as crizotinib would be a good therapy for that. So you can see here some images and tomography here, and you can see that after like the treatment was started, the lymph nodes affected was like very reduced after a few months, and after 11 totally disappeared. Therefore, like that has been like a success story into that. This is like one like brief like mentioning on one case study, but you can find more information into this on our website. Now, moving to the next part, which would be like transcriptomics. So at Novogene, we have a specific solution for like tricky samples, like highly degraded or low quality samples that especially like are treated with like the FFP derived. So what this specific kit has in like as a relevant is that it can highly cover like the expressions because of like the specific strategy on like capturing. So here in this image, you can see that like as a difference in between the normal poly-A mRNA sequencing, this strategy takes advantage of the poly-A tail to be able to capture mRNA. But instead, because of the degradation of the samples of FFP derived and other low quality samples, if like the poly-A mRNA enrichment is used, you would be missing a lot of information, and therefore instead of doing this strategy, the strategy that is called is like a probe-based capture that I will later explain to you. For this product, we take 20 days from RNA sample receival to data release, and we can provide optional bioinformatics analysis, both standard and advanced pipeline that I will explain you in a second. Finally, in this also product, we can get like high quality data. And now like going a little bit more in depth on like the possibilities of this product. So as I said before, it's especially good for like high for difficult samples that are low quality or quite degraded. It gives you like really high discovery rate for the transcriptome coding regions because of like capture technology. And also we can perform like this service with like quite low input, as low as like 200 nanograms, that it's like very here specified in this table. However, we say like the over requirements is 200 nanograms, but if the quality of the RNA is quite good, like we can go even lower. So we would assess this with like our sample quality control that also includes the DB200, which assesses the percentage of the sample that it's has like fragments over 200 base pairs. And here again, like this product has a lot of different applications, especially in cancer research and drug development. So you can see here it would be relevant for like cases of, for instance, like drug resistance, immunotherapy, gene expression, differential gene expression, cancer biomarkers, and to evaluate the microenvironment and cancer heterogeneity. As I said before, as promised, like here I show you like how good the data can we obtain for like this product. So you can see that compared with like the classical poly RNA-seq, TruSeq RNA exome has like really high percentage of clean data with very low rate, very high Q30 values, and a very low presence of our RNA. So this is also reflected more graphically here. So you can see that the error rate is kept flat along the reads, and that the clean reads percentage is really high, as this pie chart shows. And because of like the strategy of like the library preparation, most of the reads are mapped into the exon equation. So again, like just like to briefly just explain to you how this works. As I was saying, so this specific like strategy for the TruSeq exome like is using not the poly enrichment, but like using like probes that are like bioattenuated, and therefore like it can be then pulled by the structured within bits, and therefore like have this expressed part of the genome. Once more like going forward, after we do like the sequencing for your samples, we could go like through analysis, and this would be done like so from the raw data, again like doing the data quality control, doing the mapping against the reference genome using the HiStat software, and then performing the standard analysis that includes the gene expression quantification, providing you like information as these graphs for gene expression level distribution across the different samples, and information of differential expression analysis with volcano plots or like cluster analysis of differential expression genes. And finally, also going for enrichment analysis with like using gene ontology, KEK, and Reactome, and providing like some like graphs like this style with like scatter plots, and also information on protein-protein interaction analysis and functional annotation. If you want to go farther, though, we can also provide with the advanced analysis, splicing analysis, SMP and indels analysis, and also fusion analysis. So this would be like discussed with you depending on the needs of your project. Finally, and as the last two services that we offer at the clinical service, we have the pre-made libraries and long reads. And here today, I will just only briefly introduce them to you. So for pre-made libraries, is a service that is also quite used with us because it allows you like the flexibility to use a different kit that is like outside our standard offer, so that you can prepare your libraries yourself, and then we do for you like the sequencing in our NovaSeq 6,000 machines, and then provide you the raw data, or give you like some further optional analysis such as variant calling or quantification analysis. So this can be like a good alternative for you in terms of like being able to use your kit of interest, or like for solutions of like single cell RNA-seq and Syntenex. Also for the case of the long reads, this is also an option, and it's like specifically like quite relevant when there's the need of like variant discovery and validation for like some tricky and like more like obscure regions of the genome, for the detection of repeat expansions, inversions, or challenging structure variants, and also like the use of like pseudogenes and like this kind of like types of like variants. And also if you need like some methylation detection, the like long reads can be like a solution for you too, because like then by sequencing we can get like this information directly to you. So basically that would be like my last slide for this. Just want to summarize finally that I hope the message I delivered to you today is that we can offer you like a wide range of different like clinical services with the support of our like expert technical support team and skilled laboratory staff that works in certified laboratories across the globe, and we'll be providing you high quality data with a fast turnaround time and a customized safe data delivery to you. So with that, I want to finish here my presentation, and I open to any questions you might have. So thank you everyone for your attention.
Thank you, Ina, for the excellent presentation. We certainly have had some questions coming in, so I'm going to run through them with you now. So Dan has asked, when would you recommend whole exome sequencing or whole genome sequencing over the cancer panels for cancer samples?
So basically, our strategy is you would consider the gene of like the gene list of interest. So if you have like a predefined genes that you would be looking forward to check, then we'll go first into that, check whether these are included in our panels, and if not, then like we would move to more like a wider approach into like whole exome sequencing or whole genome sequencing. So like the consideration now here is that, as I said, like the most relevant genes in solid tumors are included in our NovoPM panel. But if not, then yeah, we would move towards like the whole exome sequencing and whole genome sequencing. With that, when you get to the level of whole exome sequencing or whole genome sequencing, it's important to consider which are the kind of variants that you are interested. For example, if you would be interested in structural variants, we definitely would recommend like whole genome sequencing over whole exome sequencing, because whole exome sequencing is enabled to be detecting this kind of variants. So this would be like, you know, like a further discussion depending on the needs on the project, but that would be the logical way of thinking.
Okay, thank you. Next question is from Anna, asking, what are the advantages of Twist 2.0 over the other panels?
So, well, Twist 2.2 is a very like quite not novel like panel that I was saying, and it gives you like a very like even coverage on the targeted region, and it's been proven to be like a really good solution. Also, because like the targeted region is it's a bit smaller, and with like a lower data, you can get like a quite significant depth. So that's a very good strategy. But normally, like the choice of panel, well, the whole exome sequencing kit can depend also on like previous experiments that you want to have like consistency on your data, or like the different interests, like such as like if you are interested in like mtDNA, we would maybe like recommend in our case like to go with IDT. So Twist, though, it's a very good like new panel, and it gives you like a very good coverage and evenness on the average depth sequencing depth. So yeah, that's a very good one, to be honest.
Thank you. Next is from Robin, asking, how is PacBio both highly accurate but with a high error rate? Could you explain this?
So well, PacBio has been improving like their technologies, and also like mainly like to cut like a long story short, for PacBio, what we use by basically in the end is the hi-fi reads, which are the ones that give you like high quality. So by historically like short reads like Illumina platforms, it would give you like a better like calling and very like accuracy. But also nowadays with like this hi-fi data, you can get like a very good results in terms of data and very low error rate. So yeah.
Thank you. Colin asks, what does MAF mean?
Sorry? Minor allele frequency. Yeah. Minor allele frequency. Yeah. So I mentioned before, like that's some reference sometimes that you use, like so which is the capacity of calling with for determining MAF. Yeah.
Thank you. Next is from Mike. So with 30x WGS, is the mitochondrial DNA included? Has there been any analysis on how the sequencing performs for repeats, such as for the detection of ataxia repeat expansions? It's kind of two questions in one there. Do you want me to separate them for you?
Can you go again? Like it's I missed like a bit the beginning.
So the first question is, with 30x whole genome sequencing, is the mitochondrial DNA included?
Yes, the answer is yes, because it interrogates the whole genome, so it also includes the mitochondria. Yeah.
Okay, and the second part then is asking, has there been any analysis on how the sequencing performs for coverage of repeats, such as for the detection of ataxia repeat expansions?
Okay, so You can use whole genome sequencing for this kind of approaches, such as ataxia. But like as I mentioned towards the end of my talk, for this more complex variants such as expansions, normally it's quite recommended to go to like long reads with PacBio, so that you have certainty that like you are really calculating how much these expansions have been. So having a more accurate data into that. Yeah.
Okay, thank you. Next is a question from Carol asking, can we sequence FFPE and whole tissue samples?
Yeah, sure. You can do like whole tissue, like FFPE samples. You can send us the extracted DNA directly to us, or we can perform the extraction for you, as I was mentioning before. So either way, we can accept your samples and perform the sequencing services for you. Yeah.
Thank you. Next from Simon, do you need to sequence control and tumor samples at the same depth?
No, as I was saying before, as an example, for instance, in the Novogene, so we always do, well, like the requirement for like control samples is much lower, and this would set the baseline of like the content of like the germinal mutations for the patient, for the sample, and then based on that, we would be able to be calling for the somatic mutations in the tumor sample. So tumor always like higher depth control sample can be like having like a lower sequencing depth.
Okay, thank you. Next up from Rustem asking, as researchers located in Germany, do we send the samples for whole exome sequencing and transcriptomics to the UK?
Yes. So there's no problem. We accept like samples from all across Europe, and once we receive your samples, we then like process them in our laboratories across the globe. So it can be either like processed into our UK lab or like any other accredited labs, depending on the service you require and depending on your demands or your preferences.
Lovely. Next up is from Noor, wanting to know if Novogene also provides library prep kits and gene panels for various cancer types.
So the main cancer panels that I was mentioning today in the talk is the one that we offer in our clinical service. However, if you have like a very specific interest, if it is in a small set of genes, we can always go to the targeted sequencing like strategy. But also, as I was mentioning towards the end, if you really want to do a specific kit with like a specific library preparation, there's always a possibility that you prepare like yourself the library, and then you send us the library prepared, and we proceed from that, from like library quality control until like the end, like so sequencing and providing you the data, like the raw data or the analyzed data, depending on what you need.
Thank you. So I think we'll finish with this question. So this is from Galba. Apologies if I'm not pronouncing that correctly. Firstly, saying thank you for the informative talk. So thank you, Ina. What would you suggest to a customer if they want to do genetic testing, for example, breast cancer or Down syndrome? Would that be a whole exome sequencing approach, or would you use something like Novogene? And how would the report look for such a diagnosis and the associated costs?
So you would, but you were saying like for BRCA and like Down syndrome? What?
Breast cancer in general, as opposed to BRCA, and Down syndrome is mentioned as well, yeah.
Okay. So, I mean, if you really want to have like the whole information, including like, Down syndrome information, you can go like for wider strategies such as like the whole exome or whole genome sequencing. However, if you want to like really focus into the specifics of like the mutation of the BRCA genes, Novogene panel gives you like a further like detail in terms of like higher depth into that. So it would depend a bit of the case, and if you have like, like based on like previous tests and like expectations into that. But in theory, like Novogene would be already a good solution for you.
Okay, lovely. Thank you. So as I said, I think we will wrap up there. So thank you everybody for your time today. We hope you enjoyed the webinar and found it useful. Thank you for all the questions. If we haven't managed to answer your question live, we will get back to you via email next week with the answer. In the meantime, we'll email you a link to the webinar recording and the slide deck, along with a short survey that we'd appreciate you taking the time to complete. The survey will help ensure that we can make future webinars even more relevant to you and your research goals. There will also be a link in the follow-up email for you to view a certificate of attendance if you need one. In the meantime, please stay in touch. If you have any upcoming NGS projects, you can contact us via our website form on novogene.com, and we'll put you in touch with our regional your sales representative. You can also follow us on Twitter and LinkedIn. Our website contains an archive of educational webinars, and some clinical ones that might be relevant to you are shown here. So we've got some webinars already on the clinical applications of next generation sequencing, the use of long-read sequencing technology in clinical research, and also the use of pre-made libraries for clinical sequencing. So we hope you find these useful, and we also look forward to seeing you on our next webinar, which is an exciting one. It's a webinar in collaboration with our extraction partners, BioEcho. So if you would like to join us, the registration link for that will be coming out soon. So thank you, everybody.